So many thanks for the invitation to speak today, and obviously I'm presenting, but this is a huge group effort, so thanks to all the other people that have contributed to this. I'd like to say that up front. So I would like to present, as kindly introduced, some of our recent work identifying links between the genome, the proteome, and human health. And so we know that the genome-wide association approach, or the GWAS approach, has been very successful in identifying genetic variants that are associated with phenotypes. But then the question is, how do these genetic variants influence the phenotypes? And this figure shows the omics cascade. So essentially we go from DNA in the genome, which is a template to produce mRNA molecules or transcripts. And these are translated to build protein molecules, and their activities result in the formation of metabolites, which lead to a phenotype. And in our study, we looked at how genetic variation in the genome directly influences the proteome, and how this can impact on phenotypes and disease outcomes. And so the plasma proteome, which is what we're looking at, is made up of actively secreted proteins such as coagulation factors and cytokines. It also includes products of cell leakage and turnover, an example of which is cardiac troponin, which isn't usually detected in blood, but is released when cardiac muscle is damaged, and so can be used as a biomarker for cardiac tissue damage. And it also includes soluble fragments of membrane proteins. And proteomic technologies can be broadly divided into mass spectrometry and affinity-based assays. And mass spectrometry is considered the gold standard for multiple protein detection and measurement. It digests the protein into peptides and then analyzes those using mass spectrometry. And it has the advantage of being highly specific, but has limited throughput, although I understand that it can look at many proteins, but it does also mainly cover high abundance, medium to high abundance proteins. So the proximity extension assay, which is the technology used by Olink, for example, you may have heard of, uses matched pairs of antibodies attached to unique DNA oligonucleotides for each protein. And this has increased throughput and coverage compared to mass spectrometry. And the latest platform, for example, by Olink, can measure almost 3,000 proteins. And the SomaLogic platform, or the technology, so Aptamer microarrays, they used by SomaLogic, for example, use modified DNA fragments that bind to the protein by matching the 3D shape of the protein. So in the assay that we used, there were almost 5,000 proteins, but the latest technology, the latest platform can measure almost 7,000. And we were looking at individuals from the Fenland study. We're very lucky to have access to data from the Fenland study. And this is a population-based cohort of almost 12,500 participants born between 1950 and 1975, living in and around the Cambridgeshire region in the UK. And the participants are recruited relatively young, so they were on average around 48.5 years old, which also means that they're less likely to be affected by the onset of later life diseases. Then our proteomics data, as I said, was the SomaScan assay version 4, which measures almost 5,000 proteins. And we also had data, which I'll mention, from Olink. We had 12 panels of Olink, which is just over 1,000 proteins in a small overlapping subset. And we also had some mass spectrometry data, which is around 340 proteins in collaboration with Markus Relser from Charité in Berlin. And we performed a genome proteome-wide association study. So the phenotypes were the residuals from a regression of the rank-based inverse normal transformed relative SOMA abundances. So we regressed those on, we transformed those to be normally distributed. We regressed those on age, sex, and 10 principal components, and also on sample collection site, which was a proxy for essentially sample handling. And the genetic reference, imputation reference we used was the HRC, plus the UK 10K and 1000 genomes imputation reference panel, which is also the one that's used by UK Biobank, if you're familiar with that. And we performed association analysis using the software BGenie under an additive model, and the Fenland cohort is actually genotyped using 3 different genotyping arrays. So those were analysed separately and then combined in a fixed effects meta-analysis in metal. We looked at variants with a minor allofrequency above 1%, and that left us with 10.2 million variants, including SNPs or single nucleotide polymorphisms, including the X chromosome. And so this is what's called a cis-trans plot, and it's a plot of all of the pQTLs, and on the x-axis is the position of the genetic variant, so each one of the chromosomes, and on the y-axis is the position of the protein encoding gene. And each point represents the association between the genetic variant and a protein target, and along the diagonal are, in pink, are what we call the cis variants, which are located close within 500 kb from the protein encoding gene, and in blue are the trans variants, which are further away from the protein encoding gene. And the shading indicates the significance where darker points are more significant. And so here, for example, you can see that this is a genetic variant associated with levels of, which I can't see, IL3RB, and these are the relative protein abundances for the 3 different genotype categories. And so there are just over 2,500 genomic regions associated with at least 1 out of almost 4,000 protein targets. Over 1,500 of those were in cis, so near the protein encoding gene. And at the time, almost just over 40% of those were actually unreported. So it's a massive advance on what was available at the time. And there were a total of 10,674 SNP protein target associations. The upper panel shows the number of associated protein targets for each genomic region, with circles to indicate the number of approximately independent genetic variants in the region. And we identified 8 extremely pleiotropic regions, which were associated between 59 to over 1,500 protein targets, including variants which, for example, at the CFH locus were expected to increase DNA binding affinity of circulating proteins. And so you can see these here were these sort of vertical lines where you have a genetic variant that's associated with many, many proteins. And actually the point at the top here is very small, so there aren't many independent genetic variants for that locus. And so where we have a cis pQTL, we have a SNP that's in or near the protein encoding gene. And so we can sort of understand the biology behind that, or at least we hope we can. And then we have a trans pQTL where we have a SNP that's near some other gene that's far away from the protein encoding gene. And then, however, it can also be associated with multiple proteins. And so we asked, do all of those fall into one pathway, or are they related to a single gene ontology term, or do they all belong to a community of proteins and possibly be co-regulated, or is it just unspecific pleiotropy? Are they just kind of associated with multiple unconnected different proteins? And so to do that, we constructed a data-driven protein network using Gaussian graphical modeling, and we identified 191 distinct protein communities in the network. And this is the overall protein network with the coloring for the different communities. And we then integrated this network with gene ontology mapping to try to distinguish these pathway-specific pQTLs from those which are exerting effect on multiple unrelated protein targets. And one example from the protein network that wouldn't have been identified using gene ontology alone was a cis pQTL for PNPLA3, a non-alcoholic fatty liver disease variant, which tags a missense variant that makes the protein resistant to degradation. And this leads to an accumulation of hepatic lipid droplets causing fatty liver disease. This figure shows, is a bar chart of the PQTL classification based on GO-term mapping in blue, or the community, the network community mapping in orange. And in red, in darker shades are the cis PQTLs, and in lighter shades are the trans PQTLs. And what you can see is that over 40% of the almost 5,500 independent genetic variants were actually protein specific, and almost 6% were pathway specific. And there were a number that were likely pathway specific, but there were also a large proportion that were actually unspecific. And what you can see, what this figure shows you, is that largely the cis variants, so these darker ones, were generally protein specific, whereas the trans variants were largely unspecific. We looked at the overlap between the protein targets on the version 4 SomaScan assay, and the overlap between the proteins secreted by cells, collectively known as the secretome. And what we found was that around 12% were expected to be secreted into blood, whereas almost 70% were not expected to be secreted. And there were a number that were expected to be secreted locally in target tissues, or intracellular, or bound to the membrane of cells, or secreted to an unknown location. And so what we decided to do was to systematically test for colocalization, so that's like a shared genetic signal, between our cis PQTLs, so the PQTLs that we identified near the protein encoding gene, with cis EQTLs, so gene expression, and cis SQTLs, so alternative splicing events, using GTEx version 8, which has 49 tissues, and whole blood in EQTL gen for around 30,000 individuals. And the idea was to try to pinpoint the tissue of origin. And there was strong evidence that at least half of the cis PQTLs had a shared signal with gene expression, either EQTL or SQTL, in at least one tissue. And this figure shows the protein targets ordered by the number of tissues. So the protein targets here are along the x-axis, ordered by the number of tissues for which at least one of the cis PQTLs for that protein target was also a cis EQTL, determined by statistical co-localization. For more than 70% of the cis PQTLs, the plasma protein and expression effects were directionally consistent. You can see that here. However, there were some where the protein effect was opposite to the direction for the gene expression observed across all tissues, which is actually a known phenomenon. And then when we considered alternative splicing, so this is in the lower panel here, 1/5 of cis PQTLs were shared with a cis SQTL in at least one tissue, and actually 84 were not seen when we just looked at EQTL data alone. And and so we can also use the protein data to try to identify the causal gene. So GWAS approach, as I said before, has been extremely successful in identifying genetic variants that are associated with phenotypes and diseases. But actually, the uncertainty in trying to assign the causal gene is the major limitation for being able to translate that into the clinic and experimentally validate those results. And so if we know that the variant is the cis PQTL for a gene, then we can use this to identify candidate causal genes at GWAS loci. And so we took all genome-wide significant variants from the EBI GWAS catalogue as of January this year, and we identified 558 cis regions where the PQTL was in strong LD, so highly correlated, with at least one variant for over 500 curated phenotypes. And so this figure represents the broad, in this figure, sorry, the colors represent the broad curated trait categories. which you probably can't read here, but these are the kind of different broad trait categories. And the y-axis represents the number of loci that have been reported for this particular curated phenotype. So each kind of column is one of the curated phenotypes. And then these are the number that, of loci that have been reported for that trait. And the upper panel shows the number of GWAS loci where the cis-pQTLs provided a candidate causal gene. So there previously wasn't a candidate causal gene, and using cis-pQTLs, you're able to identify candidate causal genes. And the lower panel here shows the number of loci for which the cis-pQTL refined the list of candidate causal genes at locus. So in the paper, it may have been reported that it was potentially a number of genes in a region, and using pQTL data, we were able to prioritize candidate genes. And actually, you can see here examples where the gene prioritization was facilitated through pQTL, but not through expression eQTL, are shown with a box around the outside, a border. And so there were 558 cis regions with 537 GWAS catalogue traits. And just over 24% of cis pQTLs prioritized a different gene from the reported or mapped gene in the GWAS catalogue. And we were able to, and for 14.2%, we were able to refine the candidate gene list. One example where our cis-pQTL mapping identified a biologically plausible candidate gene that wasn't implicated through eQTL mapping was for the eQTL-supported CDC-A8 locus for endometrial cancer, where we assigned R-spondin-1 as a candidate causal gene. And so this figure shows, it's essentially a regional association plot. So across the x-axis, you have the chromosomal region, where each point is association with, here we have the endometrial cancer GWAS results, where it's the minus log base 10 of the p-value. So this is the most strongly associated genetic variant. And here we have association results for the R-spondin-1 protein levels. And what you can see is that there's a high posterior probability of a shared genetic signal. So essentially the shape of the signal is very similar and the lead SNP is the same. So there's a high likelihood that the genetic signal between R-spondin-1 levels in blood and the risk of endometrial cancer are shared. As I said before, this has previously been assigned to the gene CDC-A8 based on cis-eQTLs. But obviously we were able to re-annotate it, and we see a 91% increased risk for endometrial cancer per 1 standard deviation higher genetically predicted protein levels. And actually R-spondin-1 upregulates estrogen receptor alpha, and knockout mice have an abnormal development, of reproductive tissue. So this is just one example that can illustrate the advantage of pQTL data. So then we went on to systematically assess the sharedness of gene protein disease triplets through a phenome-wide co-localization of cis-pQTL regions to create a genetically anchored map of proteins involved in common complex diseases. So essentially we took multiple phenotypes, so the summary statistics are available for all the phenotypes that we could find. And we looked to see whether there was a shared genetic signal between diseases or phenotypes and protein levels, and then created a map showing positive directions, so increased protein, increased phenotype or increased risk, and inverse directions. And so we identified 1,859 gene protein trait triplets, and so that's shown here. And there are 412 protein targets and 506 curated phenotypes. So that's a huge network. And to be able to look at it in a bit more detail, you can see here, this is the network restricted to the binary endpoints. And this figure is a summary of the summary of the results essentially. So the left panel represents the number of co-localizing protein targets for each binary outcome. So here you have the ordered binary outcomes, and then these are the number of protein targets that where there was evidence for a shared genetic signal. And you can see, for example, there were some phenotypes where we found 5 or more co-localizing cis-pQTLs, such as coronary artery disease and hyperlipidemia. And the middle panel does the same, however, it's for continuous traits, and you can see that the top phenotypes are kind of mean platelet volume and platelet distribution width, so kind of blood cell traits. And then the right panel displays the number of co-localizing phenotypes. So on the x-axis, we have the ordered protein targets, and then on the y-axis, we have the number of phenotypes that co-localize with that protein target. And one where the top one here is glucokinase regulatory protein, also GCKR. And one that we found particularly interesting was, I'll talk in a bit more. So one protein that was connected to a very large number was 37 diseases and phenotypes, was fibrillin 3. And this is an extracellular matrix glycoprotein encoded by EFEMP1. And it was associated with 37 phenotypes, including anthropometric measures, connective and soft tissue disorders, including hernia and varicose veins, and eye phenotypes. So this figure on the left here illustrates, it's another way to kind of, so rather than the stacked regional plots, this is a way to be able to look at multiple phenotypes kind of in a more compact way. So essentially, along the x-axis, we have the scaled z-score for the association between the genetic variant and the trait. So to the right here is more significant. And then the size of the point is the correlation, essentially the R squared, so the linkage is disequilibrium with the pQTL. And so if you see kind of shared genetic signal, then you expect the SNPs that are most highly correlated with the pQTL to be also most strongly associated. So there's kind of larger points to the right-hand side of the figure. Mutation, and so what we see here is that the common A allele at this pQTL is associated with lower plasma levels. So here you can see fibrillin 3. So lower plasma levels are actually associated with abnormal elastic fiber morphology. So we have lower plasma concentrations and increased risk of hernias, varicose veins, height, whole body fat mass, and hypermobility. But on the flip side of that, we also find that you have decreased risk for carpal tunnel syndrome. We also know that mutations in this gene cause a rare eye disease. And we did find evidence of sharedness of the genetic signal for this protein with vision-related phenotypes, including the use of contact lenses. Another example is at the SALT2A1 locus, where we identified a signal shared between protein levels of bile salt sulfotransferase and the risk of cholelithiasis, as well as cholecystectomy, can't talk, and increased levels of the protein are associated with increased risk of those outcomes. And we found that the signal was actually also shared with mRNA expression of SALT2A1 in the liver and plasma concentrations of multiple sulfated steroids and bile acids. And so this kind of consistent shared positive effect direction across all of these physiological entities suggests that actually SALT2A1 activity is the mode of action, where increasing protein levels results in higher levels of sulfated bile salts, and that's what increases the risk of gallstones. And actually, so this is a nice example where kind of integrating outcome phenotypes, protein data, metabolites, and expression kind of puts together this really nice picture. And so that's kind of the highlights from some initial highlights, but you can have a look for more. But then we've also made the results publicly available through an interactive web application. And so that's at omicscience.org. And many thanks to our collaborators at the Helmholtz Institute, particularly Maria and Gabby, who've helped make this available. And you can explore the targets. So there's a matrix, and you can look at all cis loci and associated aptamers. You can explore the network, and you can also do SNP lookups and download results. And so this is an example of the matrix where you can see, you can put in a target, and you can look at the number of PQTLs for the different targets. And there's also additional annotations such as UniProt ID, links to reactome pathways. So for example, here you can see the target, the full name, the protein encoding gene, the number of cis PQTLs and trans PQTLs, and you can kind of look across this entire matrix. You can also look at the network. So this shows, you can zoom in, and you can see here, this is an example for type 2 diabetes, where you can see the different phenotypes, protein targets, and directions of association, indicated by kind of solid and dashed lines. You can also do lookups by SNP and by regions. And this table shows, for example, the somomer, so the kind of the protein that you're looking at, and then the variant and the effect direction, the effect size, standard error, p-value of 0 here, but the sample size, and essentially, so you can look up all of these results. And I also just wanted to say, so I've discussed, I didn't really mention the Olink data here, but there's a recent paper published, Mike's the first author, which integrates the SomaScan and antibody-based Olink assays and looks at the phenotypic consequences of hundreds of PQTLs that were discovered for protein targets across both platforms, and kind of looking at the differences between the technologies, if you're interested. And I'd like to say a massive thank you to everyone that's made this possible, in particular Claudia, Mike and Nick, our collaborators, so Gabby, Eric, Robert, Arun, and many other people that actually haven't got their pictures here, and also colleagues from the MRC epidemiology unit, and funders and participants. Thank you. I don't know if anyone has any questions.
Yes, thank you very much, Eleanor. Thanks for the introducing this impressive piece of work that was recently published in Science. So if anybody has a question, please raise your hand so that we can unmute you. And oh, there's the first question coming in. Let me, there you go. Go ahead, Bidu.
Hi, thank you, Eleanor, for a nice presentation. I have a quick question. It's about the PQTL targets that you classify to. So do you see any association for these targets to the proteins having shared sequence homology? Do you incorporate that into your pipeline, or is that not a?
So we don't actually have this. We haven't incorporated that, no. I don't know.
So I guess the specific proteins would be just that specific protein, but not any other protein that shares the sequence homology.
Yeah, so we just have that annotation from SomaLogic, which says what the protein, so the aptamers. So we have multiple proteins, and some of those are actually targeted by multiple aptamers, but we didn't look at the shared kind of sequence homology across them. I don't know, Mike here as well. I don't know if he wants to add anything on that. But
I mean, we have some instances where multiple genes which are located close by encode for very homologous proteins, and we do see instances where it's really hard to tease out, is the aptamer actually measuring the right protein it says so, or is there some sort of like unspecific binding? So there's certainly some examples where we can't really tease this out. For the vast majority, we have seen very specific like protein encoding gene maps to what the aptamer is supposed to target. So, but there's also, I mean, we have seen cis-PTDs for 1,500 proteins, but this leaves obviously the possibility that many other aptamers may target unspecific proteins as well, and so we may miss those in the sense of these measurements are way more noisy. So this is an issue. How specific are it's the same issue with antibodies actually. How specific are they for the protein you're interested in, and at which point do you want to go for mass spec to have the actual sequences? Does this make sense? But we haven't systematically integrated this, such information other than in our like cross-platform comparison, where we really looked how many at least isoforms are there which are similar, but we haven't looked for how many off-binding effects. I mean, it's a tedious exercise for one protein. It becomes even not doable if you look at hundreds of proteins, if this makes sense.
Okay.
Thank
And
you.
with your initial list of proteins from SomaScan, do you do some like filtration to get a reduced number, or is it do you take every single proteins that you detect?
We took the full list, so SomaLogic did reannotate some of the proteins, and we removed some, but we essentially filtered down to human proteins.
Okay.
Thank you.
So there were slightly more than 5,000 on the assay, and we only included 4,790 or something.
Shira has another question.
Thank you. Really enjoyed that. Now that you gave this teaser to your other paper that you just published, comparing Olink and SomaScan, I'm just curious from that experience, if you were taking, plasma and you wanted to do a comparison with like whole exome, what would you go for between the two technologies? It sounds like you were saying they're complementary, but if you could only choose one.
You mean if you had a choice between something like SomaLogic or Olink?
Yeah,
that seems to be the question. I mean, I think that I don't know, Mike might want to comment, but I think that there are advantages and disadvantages to both, and neither's perfect. So yeah, sorry, it's not a very.
I mean, we've been asked a lot this exact question, and the usual answer, it depends on the purpose. So if you want to do something like we did, discovery, then you would obviously go for the assay with such the largest coverage. So 5,000 and now I think 7,000 is quite a good argument to find some gold nuggets, basically. Mm-hmm. There is some evidence that the level effect, like if you're really interested in protein abundances, there might be more robustly detected with Olink compared to SomaLogic, just because the nature of the assay is different. So SomaLogic is way more dependent on the conserved shape of the protein you're interested in. And if you have these common genetic missense variants or splicing variants, then your signal might even be lost using this SomaLogic assay. I mean, this could happen with all the proteomic assays, because if you have protein truncating variant, your antibody binding epitope might be disappearing as well. So it really depends on what's your purpose. Is it discovery, finding new targets? Then you probably go for the larger. If you're more onto like biomarkers you want to take forward for the clinic, then you might want to think differently. But it's really hard to make a guess, because in the end you have a disease of interest, which has a protein somewhere lying in the dark, which is only discovered by one but not the other assay. So it's hard to make a guess.
thank you.
Thank you. Are there any other questions? Otherwise, I have a couple. So while I wait for people to raise their hands, so don't be shy. And if you're too shy to raise your hand, feel free to use the chat function or the Q&A function. So now, question to Eleanor and Mike. So when I look here at the phenome-wide colocalization analysis, there was also this kind of Alzheimer's disease bubble or whatever you want to call it, or network. And I did notice, or I was a little bit surprised. I'd expected many different proteins and many more proteins. After all, kind of they have done extensive studies. They have identified plenty of SNPs also with regard to Alzheimer that are associated with Alzheimer and various GWAS studies and so on. So I was a little bit surprised to only identify, I think it's 7 proteins, and not many of them were really kind of those bona fide Alzheimer proteins. So what was your take on this?
Mike, do you want to?
I did the digging into Alzheimer actually. So yes, Alzheimer is a typical protein disease, but actually in possibly in the wrong tissue. So there have been very recent papers on brain PQTLs, brain PQTLs, so brain tissue measured proteins, and I think they found more overlap. So we're basically bound to what is leaked, secreted, or whatsoever in the plasma. I mean, we have been very lucky with many examples in the sense of that we have these cell turnover effect that we can make some interference from plasma data, but I think Alzheimer's, you may have to go for different type of fluid. And then also, maybe it's just hidden yet because there's another, so the proteomic space we looked at is 1,500 roughly, but there are obviously way more protein encoding genes which are relevant. So it could be just that we have currently still missing the genetic instruments to find the overlap with the Alzheimer's disease.
Okay, I take your argument with Alzheimer's disease and small disease of the brain then, or less reflected in blood. But how is it, for instance, you talk about, I think, 10 proteins that were associated with your with the sex difference, male, female. And when I compared that list with the proteins we normally, for instance, identify when you just look at male and female plasma, there were was very little overlap actually. And so now there you can argue again, or your argument of brain, different compartment, different organ, and the leakage doesn't necessarily apply there any longer. So let me rephrase my question. Why don't we see the bona fide sex difference, or the proteins that are bona fide sex dependent? Why don't we see those in your analysis?
And were they covered by the somologic assay?
I don't know. I didn't look at the 4,779 proteins that you cover.
I mean, I don't know. Without knowing, which ones you're thinking of, I'm not sure we can answer that. I mean, sorry, Mike,
okay.
Go ahead.
So you mean that is at the moment a little bit of a limitation of the somologic. You get what you get and you don't get upset. Basically, you see a lot, but you also miss a few.
I mean, we didn't look for sex-specific differences. We only looked at the ones that were identified kind of in the sex combined analysis here. So we looked to see whether there were any differences in effect sizes for those that we had found. But we are looking, so that's another project that someone in our group is working on to look at sex differences. But whether the ones you're thinking of were there, or I'm not sure.
Point taken. I guess I should look at the 4,700 proteins that you looked into.
I mean, you could maybe one small comment on this. You can also, I mean, I do like this idea to look more on what we would expect and what isn't there. I think it might be different for genetic effects. There has been just recently a study in the UK Biobank where they looked at a whole variety of phenotypes where we know the disease, the prevalence, incident, everything is really sex dependent. And once you go to the genetics, you hardly find anything. So genetic variants seem to be very conserved. And actually, there are very few mechanisms you could imagine how a genetic variant acts in a sex-specific manner. So, for instance, if the variant maps to a binding site for an androgen receptor or something like that, which might be very specific to either sex, but most of the things we identified seem to be conserved through the sexes, or the mechanism applies only in one. So for instance, the endometrial cancer example Eleanor showed, I mean, this is obviously a female disease, but the pQTL didn't show any evidence to be sex specific. So the mechanism by which the pQTL increases or decreases protein levels seemed to be conserved, but it happened to be that the downstream pathway, like the estrogen, the high estrogen content, is only there in females. So it looked like it's sort of more downstream that these sex affected Ps. But what we generally see is that the vast differences between males and females, what you see observationally, they're not reflected in the gene associations. Even if we really start searching for them, we don't find many of those.
Okay. And your point is well taken that I guess I took a little bit of a more too proteomic perspective on my question. So thanks for correcting me here. Are there any more questions, comments from the audience? Yes, not really. That gives me the opportunity to ask another question. So I really thought what was cool was your figure 2B and C, and you also showed it, Eleanor, this protein community analysis, basically, instead of just going by the Go annotation. Could you elaborate on that a little bit? Because I thought kind of what you showed here and your finding was really interesting, really cool, especially since that is not necessarily amenable to other approaches right now, unless I guess I go to your webpage and basically mine the data there.
I can, but I think Mike did that analysis, so I'm gonna let him.
So, I mean, I have a background in metabolomics, and I was introduced very early on with these types of metabolite networks because many of those techniques, they don't have a good coverage of the pathways. So they had to come up with some data-driven solutions. And what we've taken here forward is what others have already proposed to do on the metabolite side, but also some on the protein side. And so this network is basically generated, it's similar to a simple correlation network, apart from the fact that every correlation you would see is independent of all other proteins in the network. So that's how we try to make a hairball sort of like visible or meaningful to have. And actually many of the edges that remain really include like protein interaction partners, which is really neat to see in plasma, or like closely related in the sense of mapped to a common pathway. But then you have these examples like Eleanor showed, where a community of proteins strongly belongs together with no apparent like pathway link, but then you map on the genetic variant, which happens to be a liver variant in the sense of that it leads to sort of like liver injury in the sense of high lipid load, hepatocytes are stressed and they would start releasing their content into the plasma. And many of the proteins which were linked to this genetic variants are also fairly specifically expressed in the liver, including the biosulfatase Eleanor mentioned as well. So it provides a really neat way to also go down the biomarker route in the sense of we want to identify tissue destruction markers like cardiac troponin, or so maybe there are other proteins which have a similar diagnostic yield for a specific tissue. But this data-driven way is also a way to basically face the challenge of having plasma proteins and plasma proteins coming from everywhere of the body, basically. And it was only through this like integration with GO-term. So we clearly have examples where all the proteins which were mapped to a genetic variant fall into the GO-term, but not visible in the network. So for some reason, this relation isn't as strong in the correlation. So I think starting to integrate different sources really made the difference here. And this could obviously also be transferred to other studies in the sense of that smaller studies can map their findings onto networks derived from larger like ours or others. There are also other large scale population-based studies which then could basically team up, if you so want, to generate a data-driven backbone on pathway or network enrichment techniques.
I haven't played around with your webpage yet, but can you only kind of search one gene or one protein at a time, or can you also submit a whole list? And if then magically one of those proteins community pop up, you can see them on your hairball diagram?
I mean, currently it's really a one-by-one
Okay.
search. We haven't actually made the protein network available. So what we made available is the protein disease network. But we can certainly, if this is of interest, we can certainly make an effort to bring this up as well. So this is the entire idea, to have a living resource, which like basically survives the paper and can grow by time.
It's amazing resource that you put out there. So thank you very much. Because kind of putting a lot of stuff in a paper is one thing, but then having it as an interactive web page that can be used is another thing. So I really appreciate and kind of congratulate you to this nice web page. There's another question actually from Courtney French. Let me just unmute her. Courtney, go ahead.
Hi, yes, that was a great talk, like a wealth of information there. Going back to sort of the limitations of how you're just measuring the protein levels in plasma. And I think you mentioned there's like a brain protein data set available. I guess comparing to EQ2TL results, how correlated is it between the PQ2TLs in blood, between EQ2TLs versus in a data set in brain? And do you expect there to be huge differences between data types, the correlation between the gene expression and the protein expression? If that makes sense. Sorry.
I mean, so we looked at the brain, so EQTLs from GTEx, and largely the direction, they were kind of directionally consistent. I think something like 70% of the PQTLs and EQTLs were directionally consistent. I don't know about what that was for the brain EQTLs though. Does that answer your question? I'm not sure.
Yeah, kind of. Even maybe not specifics, but do you expect, I don't know, would it be interesting to try to do, if possible, the protein experiment in different tissues, or or for most cases or most even diseases, do you expect this mapping between the PQTLs and EQTLs to be enough to find interesting things? Yeah.
Oh, so you mean maybe if you looked at PQTLs in brain tissue or also kind of in a different tissue, would you expect the correlation to improve? I don't. Yeah. Likely, but
yeah, right.
I don't know.